Beside raw materials, if one is working with a contract manufacturer, time in the plant, the number of weeks that it takes to complete a batch is an important cost driver because that's tied both to labor and to overhead and to opportunity costs for the CDMO to have had another client come in. If the company I work for had a shorter manufacturing cycle, they could put more products through their facility and my price per batch would go down.
Hey, smart scientists! Welcome to another episode on the Smart Biotech Scientist podcast. In this episode, we are talking about process economics for antibody therapeutics. And my guest today is Brian Kelley, who is the Senior Vice President of Process Development at VIR Biotechnology. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brohlman, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics and market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech. Welcome, Brian, to the Smart Biotech Scientist podcast.
Hello, David. Looking forward to having a conversation with you today and perhaps meeting some new friends in the community if we have a chance to connect after the podcast.
Yeah, it's great to have you on today. Brian, share something that you believe about bioprocess development that most people disagree with.
That's an interesting question, David. I'd love to hear the answers from others who have participated in your podcasts. I'll begin by just mentioning I'm going to share my thoughts and positions on all the topics we're going to cover today. These don't represent any official position of the company that I work at, VIR Biotechnology. So you're getting the raw opinion here. So one area that I think is being overhyped today is the manufacturing of recombinant proteins, particularly monoclonal antibodies, using intensified manufacturing processes that oftentimes are continuous cell cultures in a perfusion reactor, sometimes connected with a continuous downstream purification train that is intended to provide a very short manufacturing window in comparison to conventional fed batch cell culture and batch purification. I see a lot of my colleagues around the industry, both industry and academia, in the last 5 to 8 years, beginning to invest in continuous manufacturing in the development phase, in the clinical phase of product development. And I have to confess, I am not attracted to the technology, having studied antibody manufacturing for quite some time now, and seeing the advances in conventional unit operations over the last 10 and 15 years, that I don't see a particular advantage. And I cannot understand what problems are intended to be solved by a continuous manufacturing process. So I'd be happy to dig into that a little bit during our conversation today. But I think that makes me a bit of an outlier in the bioprocess community. But I've had time to think about it, write a few papers, engage in dialogues at technical conferences. And so this is an open invitation for anybody on the call who's listening to connect with me afterwards. And I'd be more than happy to have a continuous discussion.
Yeah, smart biotech scientists, we'd love to hear from you what you think about this very question. Before we jump into our topic today, the process economics for antibody therapeutics, I'm curious, Brian, how did you begin your journey in biotech and how did you end up at the current role?
In a nutshell, I believe I was born a chemical engineer back in the 60s on a dairy farm in Wisconsin. I didn't know that at the time. It ended up choosing the university that was the closest to the farm, and that was the University of Wisconsin in Madison. Little did I know that it was a very strong school for both biological sciences and chemical engineering. And I began to blend both of those disciplines in my undergraduate electives, as well as the major in chemical engineering. I was probably only a junior, but still having no clue as to what I was going to do for a career, when a friend of mine says, "Hey, you're that one of those funny people that likes molecular biology, and you took that cell metabolism class in biochemistry. Do you ever think of bringing it together with engineering?" And I said, "David, no." It was David Fremont was his name, in case David's on the call. I said, "You can do that?" And he said, "Yeah, there's something called biochemical engineering. You should ask about it." Because of that great question and a friendship with Mr. Fremont, I ended up moving into biochemical engineering for grad work, and the UW school is highly regarded. I didn't realize it at the time, so that opened doors for me. And from there came probably, I'd say now, 35 years of enjoyment in the area of biochemical engineering with colleagues, classmates, coworkers through those years, even some time in academia, part-time teaching. So maybe a little bit of an unconventional path. Certainly, there are others around the industry who didn't have their career pictured in their minds when they were a youngster. But for me, I have to say it was a little vicarious, but it's been a great ride all along.
Wow, what a fascinating journey. I love that. Brian, since you have been in the industry for so long and you've seen a lot of things, I would like to know what you think are the main challenges that we as biotech scientists or engineers often encounter in process development and the manufacturing of biologics.
Main challenges, I could think of two very different ones. One is technical, and the other is cultural. On the technical side, because we are still employing a biological entity, a cell or a microbe, to manufacture our products, oftentimes there are elements of the cell biology or metabolism that are not fully understood. Perhaps for microbes that have been studied for a long period of time, all the pathways are set up and the control and regulatory mechanisms fairly understood. But for mammalian cell technology, typically employed for monoclonal antibody manufacturing, every cell line is a little different and has its own personality, if you will. And I don't believe that before I die and go to heaven, that we'll really understand all there is to know about CHO metabolism. So that's the technical answer. The cultural answer is in the companies I've worked for and others that people tell me about where they have worked or are working. Sometimes there's a lack of appreciation for the technical operations, the process development group, the manufacturing group, by virtue of a focus primarily at these companies, if they're innovator companies, on research and clinical development of protein therapeutics. And I'm not feeling disrespected, but I sometimes think there's a challenge to try to explain to colleagues who are distant from our expertise what it is we do, why we do it this way, and how it's evolved and improved over the years. And then comes the fun conversations with finance, where finance says, "Why are you spending so much money so early in this product life cycle?" And that's the nature of the beast that we deal with here. So sometimes that communication and connection to various functions around a large company, or especially a global company, can be a little bit of a challenge, I think.
I agree. I'm with you on that sometimes we can have some interesting conversations with our colleagues. And you mentioned the magic word finances. So the biotech industry has become under much pressure lately to drive down costs. So I'm excited to have this conversation now with you, Brian, about the process economics for the antibody therapeutics. Can you tell us what are the main contributing factors to the costs associated with the manufacturing of antibodies?
I'm afraid I'm going to sound like a politician, but the short answer is it depends on the product, as well as importantly, the scale of manufacturing, the volume of material I need to produce in a given year, as well as whether one is in a clinical stage of development, making just a few batches per year to support phase 1, 2, or 3 trials, or whether you're in the commercial phase where you may have a commercial product that commands regular manufacturing campaigns, or even a blockbuster where you may be challenged to supply enough material for the market. So the breakdown of the process economics in all of those combinations leads to I can generalize, but there's really no crisp answer to that. But as I think about the late stage clinical and commercial stages, very large scale manufacturing potentially, the raw material costs can be significant. The consumables used for each batch, production media for CHO cell culture, can be quite expensive if it's commercially proprietary media from one vendor. That could be sometimes the largest driver in dollars per gram, the cost of goods for a product. The capture of most monoclonals takes place on a protein A column, and we all know how expensive protein A resins are per liter. They can be reused many, many times, but that cost in a clinical phase is really high. And it has driven some groups to try to find an alternative to protein A that has not really been successful. It continues to be the workhorse in the industry, but multiple vendors are now producing protein A resins, and so there's some more competition than there was 20 years ago. Another raw material just closes out quickly that may not be obvious as a cost driver in purification is the virus removal filtration step. These virus removal filters are only single use. They cannot be cleaned and reused. And so the cost per gram purified is a strong function of the capacity in grams per meter squared of the membrane area. The prices of the membranes themselves are pretty expensive. Beside raw materials, if one is working with a contract manufacturer, time in the plant, the number of weeks that it takes to complete a batch, is an important cost driver because that's tied both to labor and to overhead and to opportunity costs for the CDMO to have had another client come in. If the company I work for had a shorter manufacturing cycle, they could put more products through their facility and my price per batch would go down. So it's an interesting driver to recognize for those of us who are working with contract manufacturers and not managing an internal asset or a network that some companies have established for antibody manufacturing. There aren't a lot of good papers on cost of goods that I would trust. All due respect to our academic community, I put a little bit of a discount on an academic estimate of the cost of goods to manufacture. I recognize the models they're using. They're doing the best they can, but some of the real numbers, the inputs of the raw material costs, for instance, are not well understood and vary from large company procuring 1 million liters of media a year to a smaller company that just uses a catalog price for that media as an example.
Well, this can be a significant issue, especially for a smaller company when you just have a few clinical batches to produce. How do you strike now a balance between, obviously, yes, you want to have a high quality, you want to be fast, you want to bring your therapeutic to the clinic as fast as possible, but still be as cost effective as possible? How do you navigate that tension?
The quality first, indeed, quality is first, will be driven by your ability to manage internal assets and the compliance and operations execution and success of the runs. If that's in your control, it should be a major focus for you because, as you just mentioned, speed to clinic is really important. And the first clinical batch for phase 1 studies, and even before that in the product lifecycle, the production of material for a tox study, a preclinical GLP tox to file an IND is oftentimes critical path. The external community and the CDMOs that some companies will use for clinical development are, in my view, for monoclonal antibody development and partnering. It's a fairly level playing field. There are a number of global and domestic CDMOs that can work at smaller scales of operation. Some can jump right to 2,000 liters or 4,000 or 5,000 liter reactors for early phase work. And so the technology is a level playing field. But the compliance and quality that are enjoyed with each CDMO is something you'll want to monitor very closely and look for trends if you work with a CMO for more than one project or over more than 1 year. Because depending on a number of factors, there can be ups and downs with regards to the focus on quality. And it's not typically product quality that I would worry about. It would be the compliance and documentation, perhaps, or the ability of a CDMO to successfully pass an inspection by a health authority for the clinical or commercial phase of manufacturing. So I'd like to go fast. I'd like to have a high probability of technical success with the CDMO, for instance, but I also want that very high quality and assurance of no regulatory risk associated with that manufacturing and production. I guess I'm asking for it all. Maybe the fourth vector is the cost of the contract. And sometimes you might get what you pay for. And so keeping an eye on the reputation of a firm that you're working with and feedback your own learnings to them so they can improve is a real important part of a partnership between an innovator company and a CDMO, I think.
Hey, smart biotech scientists. I hope you've enjoyed our conversation with Brian Kelley, who is the Senior Vice President of Process Development at VIR Biotechnology. Coming up in part 2 that will drop this Thursday, we are talking about innovations and the next chapter for antibodies. And perhaps you are wondering how you can take your process economics to the next level. So that's why I offer you a free 30-minute assessment to talk about your challenges and help you get started so that you can make your process development a success. Just use the link in the show notes, and I'm looking forward to speaking to you on the call. All right, smart scientists. That's all for today on the Smart Biotech Scientist podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.